everyone, this is your educator Priyanka Thakur. I'm teaching you life sciences and welcome you all on our YouTube channel Gurmantra Shiksha Ka Granth. So this is your phytohormone part 5 in which we are going to know about the signaling pathway of Auxin and Gibberellin. I have already covered the signaling pathway of ethylene when I taught you ethylene along with that along with the biosynthesis and physiological effects of ethylene. I also told you about its signaling pathway. Today I will be covering the signaling pathway of Auxin and Gibberellin, and in the next lecture I will be covering the signaling pathway of cytokinin and abscisic acid. Okay, I hope you are enjoying these videos. So do you want to hit the like button? No, that's all right. Let's move to our signaling pathways and we will start with the Auxin signaling pathway. Remember one thing, when you are studying these pathways, you will be seeing some terms like ligand, receptors, like repressor, transcription factors, responsive elements, responsive genes. These terms will be common to all the different pathways of different hormones. Okay, different hormones signaling pathways will include all these terms, but like I'm saying ligand, but ligand will be different for a Auxin signaling pathway. It will be different for a gibberellin or abscisic acid signaling pathway. Just like that, receptors, repressors and transcription activators or responsive elements, these are also be different for different hormones. Okay, so that let's start with our Auxin signaling pathway. As the name indicate, it's Auxin signaling pathway, so ligand is going to be Auxin itself. Auxin is a ligand in Auxin signaling pathway. Okay, so see, receptors. Receptor is TIR1 transport inhibitor response 1. Okay, also known as SCF-TIR1-AFB. In many of the books you will see it is written like SCF-TIR1-AFB. Why? I'll tell you the reason. For now, just know that TIR1 is an intracellular receptor. And why it is also known as SCF-TIR1-AFB? Because it acts as a component of SCF having E3 ubiquitin ligase. What does it mean? It means that TIR1 is actually a F-box protein. Okay, it is a F-box protein, and F-box is itself a component of SCF complex, which is SPK1 culin F-box protein complex. Okay, what is SCF? It is SPK1 culin F-box protein complex, and TIR1 is a large family of there is a large family of F-box protein, and TIR1 is also a F-box protein. That's why it can be written as SCF-TIR1-AFB, an intracellular receptor protein. Okay, you can write it just like this, SCF-TIR1-AFB protein. Fine. Now all F-box protein have E3 ubiquitin ligase activity. Okay, it can be E3, even E2, anything, but this one has E3 ubiquitin ligase activity. What does it mean? It means that receptor of auxin can bind to auxin, as well as it can cause ubiquitination of other proteins. And after ubiquitination, those proteins are targeted to proteasome, where those proteins are degraded in that particular proteasome. Clear? So the receptor of auxin is performing actually dual function. It is acting as a receptor, but it can also it can also cause ubiquitination of proteins. Okay, so this property of ubiquitination plays an important role in the signaling pathway of auxin. So now we know that TIR1 is a F-box protein. There is another receptor for auxin, which is known as ABP1, auxin binding protein 1. This ABP1 is located in endoplasmic reticulum and plasma membrane. Okay, see, Auxin binding protein 1 is the receptor. I have told you during the polar transport of auxin that how the cell wall is acidified and what is the role of proton pumps. Okay, so those particular function like cell wall acidification and proton pump functions are under the control of this receptor of auxin, but we do not know much about this receptor, so we won't discuss it in detail. But you should remember that auxin binding protein 1 is also a receptor of auxin. Main is TIR1, which is transport inhibitor response 1. Okay, so as F-box receptors like F-box proteins are found in cytoplasm and nucleus, and we know that transport inhibitor response 1, that is TIR1, is also a F-box protein, and it is an intracellular receptor. So it is very clear to us that it is found in within the cell, and it is a F-box protein, and F-box proteins are found in the cytoplasm and nucleus. Here comes the villain now. It is about the hero. Now it is about the villain now. Repressor inhibitor is the villain, which is AuxIAA in case of auxin signaling pathway. Repressor or inhibitor in case of auxin signaling pathway is AuxIAA. And transcription activator, there are also some transcription activator which are auxin response factors. Okay, transcription activators, or you can say transcription factors. Okay, these are the transcription activators. Auxin response, these are auxin response factors. Okay, you will see auxin response elements. This auxin response factor will be acting upon these auxin response element, which then be able to cause the transcription of auxin responsive genes. Okay, so this is auxin response element which possess a conserved sequence 5 in TGT CTC in 5 prime to 3 prime direction. If you are reading it in 3 prime to 5 prime direction, then it is CTC TGT. Everything is clear now. So what is the ligand in auxin signaling pathway? It's auxin itself, and the receptor is transport inhibitor response 1, also known as SCFTIR1 AFP, which is a F-box protein. F-box is a component of SCF complex, and it is having E3 ubiquitin ligase activity. It is an intracellular protein. Okay, and the other receptor of auxin is ABP1, and repressor is AuxIAA. Transcription activator is auxin response factor, which will act upon auxin response element having a conserved sequence TGT CTC in 5 prime to 3 prime direction. Now you know everything, so we will move on to our not Gibberellin, I'm sorry, I apologize for this. Okay, for our auxin signaling pathway. Okay, so what happens in the absence of auxin and what will happen in the presence of auxin? These we will study this in each and every signaling pathway. You will study what will happen in the absence of the phytohormone, particular phytohormone, and what is going to happen in the presence of that particular phytohormone. So we will know here what will happen in the absence of auxin first, and then we will go to know what is happening in the presence of auxin. This is your repressor. These are the auxin response gene, and this is auxin response element. But on auxin response element is attached our auxin response factor, but it is repressed by auxin repressor. In the absence of auxin, auxin repressor represses the auxin response factor, which then fails to activate auxin response element. So there will be no response of auxin responsive genes. You get it? No auxin means no response of auxin response gene. Why? Because in the absence of auxin, the repressor is acting on auxin response factors. When these factors are inactive, then auxin response elements won't be able to perform its function, and it won't be able to activate the transcription of auxin response gene. So this is all about the auxin signaling in absence of auxin. Actually, there is no response of auxin genes in the absence of auxin due to this AuxIAA pressure, which is binding to the auxin activator protein. What happens in the presence of auxin? See, In the presence of auxin, this auxin repressor will be removed. How? See, these are our auxin response gene, similar setup. But then there is the entry of auxin and it binds to its receptor SCFTIR1AFB. Auxin has the ability to recognize this AuxIAA repression. So auxin will recognize the AuxIAA repression, and as you know, receptor has the ability to cause ubiquitination of a protein and to target it to the 26S proteasome for the degradation. So auxin will recognize AuxIAA repressor, and there will be the formation of then receptor repressor heterodimer. Okay, this is the repressor, this is the receptor. So there will be the formation of receptor repressor heterodimer. Now this receptor will cause the ubiquitination of this repressor. After ubiquitination, it will be targeted to 26S proteasome, where it will be degraded. Now the receptor has been degraded and our auxin response factors are can be activated. These are free to perform their function now. There are number of auxin response factors in the cell. Okay, so for effective response, there might be a dimer. Okay, so now auxin IAA has left its place, so next ARF can join the previous one and form a dimer. Now it will result in the effective activation of auxin response elements that will result in a perfect transcription of auxin response genes. So this is all about auxin signaling pathway. I hope everything is clear to you now. Okay, is it clear now? In the absence of auxin, repressor is binding to the activation factor. Activation factor fails to perform its function, so all other function will be halted, like auxin response genes will not be able to perform their function. But when auxin is present, auxin will recognize the repressor and its receptor will form a heterodimer with the repressor. Receptor can cause the ubiquitination of the repressor. After ubiquitination, it has been ubiquitinated, then it will be targeted to the 26S proteasome and the repressor will be degraded. Now there is no repressor, ARF can bind to the another ARF, it will form a dimer, and now effective transcription of auxin response element genes will take place. So that is all about auxin signaling pathway. Now let's talk about gibberellin signaling pathway. In gibberellin signaling pathway, gibberellin is the ligand. Receptor is your GID, GA insensitive dwarf 1. And repressor is DELLA proteins. DELLA proteins play a major role in gibberellin signaling pathway. These proteins are actually nuclear proteins, and these proteins act as a repressor. We know that DELLA proteins are repressor. Why DELLA proteins are named so? Because they have a conserved sequence: aspartate, glutamate, leucine, leucine, alanine. And you know that alphabetically these are represented as DELLA. Okay, glutamate means E. So that's why DELLA proteins are named as DELLA proteins. DELLA protein possess two domain. One is DELLA domain, another one is GRAS domain. DELLA domain will be responsible for the inhibition of a number of function of different factors. Okay, but GRAS domain will bind to receptor ligand complex. Okay, GRAS domain of DELLA proteins bind to receptor, or I can say receptor ligand complex. In rice, DELLA protein is named as SLR1. Sometimes they can ask you, SLR1 is a DELLA protein. Maximum of the times the question is asked by the name of DELLA protein itself. But if they ask you SLR1, what is GAI, RGA? These are the names of DELLA protein. SLR1 is a DELLA protein in rice, and GAI and RGA are the DELLA proteins in Arabidopsis. Clear? Now, let's know about what are the targets of DELLA proteins. DELLA targets are PIFs, phytochrome interacting factors. These phytochrome interacting factors regulate the elongation process in the presence of gibberellin, okay, gibberellic acid. When gibberellic acid is present, DELLA proteins are degraded. Remember this, there will not be any functioning of DELLA protein in presence of gibberellic acid. So at that time, when gibberellic acid or gibberellins are present, phytochrome interacting factors will be able to perform their function effectively, and they will be regulating the elongation. How DELLA proteins in the absence of gibberellic acid can cause inactivation of phytochrome interacting factors. Actually, these DELLA proteins prevent the phytochrome interacting factors to bind with DNA, or these DELLA protein can also cause the degradation of this phytochrome interacting factors. Is it clear that what is the role of DELLA proteins? DELLA protein can cause the degradation of phytochrome interacting factors, or these proteins can also prevent these factors to get attached to the DNA. They prevent their attachment to the DNA. Next target of DELLA protein is pre-folding or molecular chaperones. Pre-folding or molecular chaperones are found in cytosol as well as in nucleus. We know that protein pre-folding or chaperones are responsible for the protein folding, and mainly they are going to play a role in the folding of protein beta tubulin, but these DELLA protein restrict these chaperones to the nucleus, and we know that receptor of gibberellin signaling pathway is GID, which is gibberellic acid insensitive dwarf 1. So we know all the components of gibberellin signaling pathway now, and we will talk about how gibberellin responsive genes are going to perform their function, and so that all the function which gibberellin is going to perform will be regulated. Okay, so remember, PIFs and molecular chaperones are the DELLA targets, and I also told you how DELLA proteins are going to prevent their functioning and what will happen in the presence of gibberellic acid. Okay, in DELLA protein in rice is SLR1, in Arabidopsis it's GAI or RGA. The two domains in DELLA protein is DELLA domain and the GRAS domain. Okay, fine. Now see what happened in the absence of gibberellin. If there is no gibberellin, then receptor will not be able to recognize DELLA proteins, and DELLA proteins will be inactivating the transcription factors. How they will inactivate the transcription factor? Either they will prevent their binding to DNA, or they will cause their degradation. And if no transcription factors are activated, there will not be any expression of gibberellin responsive genes. You understand? No gibberellin means no recognition of DELLA, no binding to receptor won't be able to recognize DELLA, then DELLA is free to perform its function. It will be preventing the binding of transcription factor to the DNA, or it will cause their degradation. And if transcription factors won't be able to perform their function, then there will be no transcription of gibberellin responsive genes. Got it? This is plasma membrane. PM doesn't mean prime minister. Okay, it is plasma membrane. Oh ma'am, we are also having the common sense. We are a life sciences student. At least we know this thing. Okay, great. Okay, so this is gibberellin. Gibberellin will then bind with its receptor, as we read in Auxin receptor, that is a ab-box protein. GID1 is also a ab-box protein. It is also a component of SCF. Okay. Now, gibberellin will recognize DELLA protein, and GID1 will cause ubiquitination of DELLA proteins, which then will be targeted to 26S proteasome and will be degraded. When DELLA proteins are degraded, transcription factors are now active, and gibberellin responsive genes will be able to be transcribed. Okay, they will be able to give their expression, and we will get the expression of gibberellin responsive genes. So you understand, in the absence of gibberellin, GID1, the receptor will not be able to recognize DELLA, and DELLA will be freely performing their function of inactivating the transcription factor, and there will be no expression of Gibberellin responsive gene. But when Gibberellin is present, the Gibberellin is going to recognize the DELLA protein, and GID1 will bind to the DELLA proteins, and because it is a F-box protein, it will cause the ubiquitination and then degradation of the DELLA protein. When DELLA protein has been degraded, transcription factors are now activated. Transcription factors are now activated, then there will be the expression of Gibberellin responsive genes. This is all about the signaling pathway of auxin and not cytokinin, it's Gibberellin. And I hope everything is very clear to you without any doubt. Are you having any doubt? You can ask me in the comment section. If you want, you can share the video. If you like it, then do share the video for me. Okay, do it, not for yourselves. Okay, for me, just do it if you want to. Otherwise, it's all right. But before leaving, let me tell you, if you want to prepare for CSIR net, like June 2021 CSIR, you can join Unacademy. You can take the subscription of Unacademy, and on your screen you have detail of your paid classes. Neha Tanisha ma'am is a very smart educator on Unacademy, so you also need to be smart. Do not pay the full fee. You can use the referral code Neha to get the 10% off. You get it? Okay. So you can avail a number of classes on Unacademy along with Neha Tanisha ma'am. There will be other educator also. In this fee, you will be able to avail all the educators classes. Okay, so don't worry about anything. Don't worry, like, don't panic like, oh, we will get or the only we will get classes from only one educator in this amount. No, you will get classes from a number of educators, like whatever number of educator is there. But you have to you definitely have to follow Neha Tanisha ma'am, and don't use any other referral code other than Neha, which is a referral code of Neha Tanisha ma'am. Along with this, do not forget to press the bell icon, and means first subscribe to our channel and then press the bell icon to get the notification. And you can check out the description box in which you will get the links to different platforms of Gurmantra. Share your love on us and we'll share our love on you. So that's all for today. Take care. Stay safe.